would like to welcome Julie Lewis.
Thanks, Cindy, for the good introduction. Hi, everyone. Like she said, my name is Julie Lewis, and I'm a PhD student at University de Moncton, which is a French-speaking university, and Mount Allison University, an English-speaking university, so I'm really bilingual. And both universities are located in New Brunswick, Canada. It's my first time being here in Texas, so I'm really glad I was invited to this amazing conference to present my work. I only started my research project a year ago. In fact, it will be a year in May, actually. So I will only be presenting preliminary findings on bacterial co-infections I detected in Morgellons and Lyme patients. Before my PhD, like she said, I was working on Lyme disease, so it's really nice to have a background that helps me with my current research project. Generally, ticks can be infected with and transmit multiple microorganisms, some that are pathogenic, some that are not. In the table over there, I don't know if I can point from here, we have data from a microbiome study by Sperling and colleagues at the University of Alberta in Canada. They looked at microorganisms that were infecting several ticks, and you see them across the table. My supervisor referred to this tick that I've highlighted in red as the evil tick, since it's been shown to be infected with almost all the microorganisms that are on that list. There are really big numbers for each and every one of them if you don't see really good what's going on there. Whereas the other ticks are infected with sometimes only a few of them. So it really depends on the tick. This tick here was actually from our collection at Mount Allison University. We gave them to analyze, and it was found feeding on a dog in Nova Scotia, so a province near New Brunswick. Poor dog. I don't know if he transmitted all of that, but I don't know how sick the dog was, but it's not good. Ticks can transmit many diseases. Example of these, we've heard them: Lyme disease, of course, Rocky Spotted Mountain Fever, babesiosis, ehrlichiosis, anaplasmosis, but we haven't heard tularemia, Colorado tick fever, tick-borne relapsing fever, Powassan disease. So it's a very extensive list that, of disease that can be transmitted by ticks. Now let's talk about the bacteria of interest for my research project before we start discussing Morgellons. Borrelia is a spirochete bacteria, meaning that it has a corkscrew shape with a longitudinal endoflagella that runs along its whole body, as you can see in the picture right here. This allows it to dig through tissue and swim in blood. However, Borrelia can take other forms. It's pleomorphic, so especially under unfavorable condition, it can take these resistant forms, which are called cysts. And they do that especially, like, for example, during antibiotic treatment. Borrelia is responsible for Lyme disease and relapsing fever, as we'll see in my next slide. What we call Borrelia burgdorferi is, terminologically speaking, actually known as Borrelia burgdorferi sensus stricto, whereas the Borrelia burgdorferi sensus lato, highlighted here in red, includes all of these species, including the Borrelia burgdorferi sensus stricto. At one point during its evolution, Borrelia differentiated into the Lyme group and the relapsing fever group. Rickettsia is an obligated endobacteria, which means it can only live and replicate inside of their host cell. It is transmitted by ticks, but also other arthropods, such as fleas and lice. It causes different types of fever with rashes or lesions, and this is all around the world. In fact, typhus was really well known back then, and it has been called the second, what's the name in English? Pest in French. So it's been, it was really a big thing when the sanitation conditions were not good, because it was transmitted by lice and fleas a lot back then. Plague, is it? And so we will talk more about these bacteria once I get to the part of my presentation where I talk about my research project. As you all know, Morgellons is a controversial, debilitating condition with an impressive list of multisystemic symptoms. Dermatologic symptoms include painful and itchy skin, spontaneously emerging, slow-healing skin lesions, and embedded microscopic multicolored filament that sheds from the skin. So we can see them at 100 magnification, magnified 100 times here. There are many more other symptoms, and they vary from patient to patient to make things even more complicated, like we needed that. Morgellon is found worldwide, but unfortunately, because it is so understudied, there's not very much literature on its prevalence. The only distribution map I could find was with families registered with Morgellon Research Foundation in the US. And this was in 2007, so more than likely it increased since then. The states with the more cases were California in first place, Texas, and Florida. I didn't find much information on the prevalence for Canada or elsewhere in the world, for that matter. And almost everybody I talked to about Morgellons in Canada had never heard of it. So I'm trying to change that by educating people over there. Thank you. There have been a few hypotheses to explain the cause of Morgellons. And one is starting to be more recognized, as you know by now, as the body of evidence that we have grows. It is the most important hypothesis for my project. It is the biological hypothesis where Morgellon has been associated with an atypical production of endogenous filaments and a Borrelia infection. It has been suggested that they could be transmitted by ticks. As you have seen most of these pictures yesterday, I will go through this quickly. So keratin and collagen stains showed filaments that are biologic in origin. In the first picture, we can see a section callus of a Morgellon patient stained with keratin. So the keratin shows in brown, and that's magnified 100 times. The next picture over is section callus stained with the Gomori trichome stain, which stains keratin in red and collagen in green, and that's also at 100 times magnification. Last picture is a longitudinal section of the filament, and again, keratin is shown in brown. And this is a little bit more close-up than the other pictures. For the association between Morgellons and Borrelia, the first two pictures are epifluorescence picture, which I also did for my research project. I'll show you that after. The first one is a close-up of the bacteria, 1,000 times magnified with oil immersion. And then the other one over is callus, and what is great about that one compared to the other one is you can see how many Borrelias are lighting up in this section of the callus, but you need to go at a lower magnification to see that. And then the other one, also 1,000 times magnified, so you can really see the shape of the bacteria a little bit more, is done by immunostaining with an antibody that is against Borrelia. Other bacteria associated with Morgellons include, as you see, and I'll just name a few here, other species of Borrelia, Treponema denticola, Bartonella, H. pylori, and Agrobacterium. So far, I tried 3 of them, to find 3 of them, and also other ones that are not on this list, and I will show you the results after. Before we start discussing my research project, we need to discuss bacterial-host interaction a little bit. First, what is epigenetics? Well, it's how the environment can be affected, can affect which genes is turned on and off. Thus, it ensures the manufacturing of proteins according to what is needed by the organism in that moment. It's something that's really dynamic. It constantly changes to respond to the environment. So epigenetic is nothing more than functional groups such as methyl and acetyl, for example, that are added to DNA or proteins that are wrapping the DNA, which we call it histone. And that loosens the DNA so that the gene is available for expression, meaning that it will make the protein that's on that gene. And you can see this on the picture right here. So you have the DNA that is starting to wind up, and then you can see it's wrapped around those histone, and you can see the methyl group that's been added right here to the DNA. And then the last picture in that diagram there, you see that the DNA is really loose. So that's what I was saying when you want to express the gene, there needs to be some room there so that the protein can go and produce that protein. The diagram I have there shows the complex interactions between the vector, the host, and the pathogen. Once inside the host cell, endobacteria are known to epigenetically modulate host cells in numerous ways, and we have examples from that in the literature. I'll just go through a couple of them just to show you, but there's a lot more. Borrelia-induced dermal filaments, rickettsial infection prompted histone deacetylation in ticks, and Bartonella changed the expression of the human host genes involved in many things like immunity, cell cycle, angiogenesis, and reproduction. So with epigenetic, the endobacteria can really control the host cell to make it do what it wants. Enough with the background, it's time to talk about my research. Since I started learning about Morgellons, I've been interested in finding out why some patients have Lyme, some have Morgellons, while others have both. Our hypothesis postulates that Morgellon patients with Lyme disease have a co-infection of Borrelia and another endobacteria, like Rickettsia, for example, and they interact together to epigenetically reprogram fibroblasts and keratinocytes to abnormally produce Morgellon filaments and, well, the disease. These are real cells that I took a picture of them during my cell culture training at the Atlantic Cancer Research Institute. So, can see what it looks like. And this will be useful for my cell culture training that I learned there. Will be really useful for the next part of my research project, and I'll talk to you about that later at the end of my presentation. I'll go into the methods a little bit more than my fellow presenters. So let's begin with the sample collection and the cultures. Clinical partners were collected our samples for us in Alberta, Canada. Samples were of various types, so there's skin, blood, knee and ankle aspirate, urine, periodontal, and genital. Samples were then cultured in BSK medium, and the DNA was extracted. BSK medium is the standard medium used for the culture of Borrelia. Oh, and there's a picture of my culture. And you see at the bottom, the little white part, that's all the cells that fell to the bottom, precipitated. Primers are small pieces of DNA that we use to amplify a targeted fragment of DNA in PCR. Most of the primers I tested were from the literature, but I designed a few of them myself manually using these online website, and program. So to do this, I first had to align the genes of different species. So I did that for Borrelia and for Rickettsia. Then I used OligoCalc, which is a website that anybody can access. And I checked for problems with my primers using that. So examples of problems that can arise with primers. So the primer can fold on itself and bind to itself, which is not really useful because then it will not bind to the DNA. And it can also bind, because for PCR, we have a forward and a reverse primer. This is how it amplifies just a section of DNA between those two primers. And the forward and the reverse primer can bind to itself, and then it wouldn't be able to bind to the DNA. So these are problems you want to avoid. Then I tested my primers using this program that does PCR simulation. This program is called AmplifyX. So it's like a little pre-test before I actually get to use them. Nested PCR is a molecular technique that allows the detection of a small amount of DNA, such as arises from a bacterial infection in a host. It involves a first round of PCR using an outer primer pair, and then the samples are prepared with that and put in the thermocycler, which is just basically a machine that heats up and cools down as DNA is amplified exponentially. The product of this first round is then used in a second round of PCR using inner primers that are nested inside of the outer primers. So it will make a smaller amplicon from the bigger one that I got in the first round. Amplified sequence were then confirmed by sequencing, which is just a read of the DNA. This is the result we get back from sequencing. The peaks of different colors represent different bases of DNA. And these are the 4 bases of DNA, so it will just be a read of each of those letters. That's why you see the T, C, A, T, C, T, G, G as the peaks form. Then we input that sequence into a website called BLAST, which stands for Basic Local Alignment Search Tools. It gives us a list of organisms that matches more closely to our sequence by aligning it to sequences from its databank. I also performed FISH, which stands for fluorescence in situ hybridization. For that, we use beacon probe, which are designed against the flagellin B gene, so that endoflagella that I said that was on the bacteria, that's the gene we're targeting. To do this, I dehydrated the tissue, I embedded it in paraffin, and then I sectioned the scabs, the embedded scabs, 10 microns thick. With the support of paraffin, it allowed me to cut really thin slices. Cultures were acetone fixed on chart slides, and both of them, the scab and the culture, were stained with beacon probes. Oh, that's the chart slide. A beacon probe is a primer on which we added a quencher and a fluorochrome. When the molecular beacon is not bound to the DNA, like we see in diagram A, it does not fluoresce because with the primer folding on itself like that, the quencher is close to the fluorochrome and it absorbs the fluorescence of the fluorochrome, so we don't see any light. On the other hand, when the molecular beacon is bound to DNA, it fluoresces because the quencher and the fluorochrome are far enough from each other that the quencher cannot absorb the fluorescence emitted by the fluorochrome, as you see in part B of this diagram. Beacon fish slides were then imaged on an epifluorescence microscope. The microscope has a mercury lamp, which is considered to be the best illumination source for epifluorescence. This light source is at the back of the microscope and is directed onto the sample through a filter that is suited for the wanted fluorochrome that you're using. We use the fluorochrome FITC, which appears green, as we'll see in my pictures, and that's the same fluorochrome also that Middelveen and colleagues used for their picture of a fish. Immunostaining is done using an antibody that's against Borrelia. That antibody, so that's the antibody there that will bind to the antigen of Borrelia. That antibody is linked to HRP, which is oxidase peroxidase, horseradish peroxidase, sorry, and then we add this TMB there, which is a long, complicated name too, and there's an enzymatic reaction, and it creates this brown-colored product, so we're able to actually see it under a light microscope. From all of the primers I tested that I showed you earlier, I was only able to find one for Borrelia and one for Rickettsia, so I agree, it's not easy. You need to try a lot of things to see what will work. And yeah, only one for Borrelia and one for Rickettsia produced sequenceable result that we could use. Once PCR is done, we run it through a gel by applying a power to separate the fragments of DNA based on their sizes. This is how we can tell if there's Borrelia DNA amongst all of that other DNA. Here we see pictures of my gels where I found Borrelia and Rickettsia. And then sequencing and a bioinformatic analysis confirmed that I have amplified Borrelia and Rickettsia. Morgellons in Lyme patients were found to be infected with several Borrelia burgdorferi strains and isolates, as well as an uncultured Rickettsia species. When grouped by samples instead of patients, because sometimes we had more than one sample type for patients, for several patients, my results indicated that 2 Morgellons patients were co-infected with Borrelia and Rickettsia, whereas no co-infection was found in Lyme patients. Like I said, this is preliminary results, and we'll discuss that a bit more in the strengths and limitation part of my presentation once I get there. Now for the results of my fish for Borrelia. All of these pictures that I will show you have been magnified 400 times. This first picture here is my positive control. So we just used a culture of Borrelia burgdorferi strain B31 that we bought. And so we're supposed to see Borrelia, in there. And I don't know if you can see really well from there, but I saw it really well on my computer. There's a couple of bright spots, really brightest than everything else, and this is Borrelia. We think it's Borrelia. Next, these are my positive control. The first one is for culture, the second one is for scabs. So we don't see anything as bright as the positive control here. So there's no Borrelia in these samples here, but these 3 at the bottom are my samples that I tested. The first 2 being for Morgellons, the last one being from Lyme disease patient. The first picture at the bottom is a culture, the second one is a scab. And the Lyme one, the last one, is a culture also. There was, especially in the first picture at the bottom, there was a really nice spirochete looking shape that I could see that it really light up bright. I don't know if it shows a lot there because I waited too long to take the picture. And if you wait too long, it will do what it's called a photo bleaching. It'll start to disappear, so you got to be really quick in taking these pictures. But I will get more practice at doing this, and I was thinking too of being trained to do the oil immersion, because then I could magnify it even more, and we could see the shape better that way too. The week before I came here, I started doing immunostaining for Borrelia, but I didn't detect Borrelia, not even the positive control. So I still need to optimize. I will practice my technique, tweak the protocol a little bit, maybe use a primary and secondary antibody instead of just having the one that I showed you in the immunostaining picture before. And that will allow us to get a stronger signal. I won't be able to do this for Rickettsia because there's no available antibody for Rickettsia, but at least I can do it with fish once I find primers that really work good. To summarize, Borrelia was found in Morgellons patients, so this further supports or strengthens the findings found by Middleveen and colleagues. My study also revealed the presence of Rickettsia found in Morgellons patients, so we need to update the list of bacteria associated with Morgellons. Bartonella was not detected. It might not grow in BSK medium, but if patient would have been infected, Bartonella DNA would have been present. I don't know if it would have been in enough to be detected with nested PCR, although it's a really sensitive method. But regardless whether it grew in the culture or medium, we would have been able to find it. But maybe it's just my primers too. I tried a lot of Borrelia burgdorferi primers, and I tried 2 for Bartonella. So I will keep trying to find this, and we will try PCRing the blood samples too. For Borrelia, I found it using different techniques, so PCR and sequencing, as well as the probe. Not immunostaining yet, but hopefully I will be able to add that pretty soon to the list. The hypothesis of the research project is supported so far, so that was the co-infection between Borrelia and Endobacteria. And the strengths and limitations of the study, for the strengths, there was biological replicates for certain patients. So we had more than one type of sample for some of these patients. Another strength is that we use different methods, and we have used and will use different techniques. So we already did DNA in situ with the fish, and then we have a series of in vitro experiments that will be really exciting, I think, and I will get to work on that soon when I come back to Canada. Limitations of the study, well, just like any other techniques, it's not perfect, so there are false negative and false positive. I also have a small sample size, but I only started not too long ago, and I have a lot more to test, so this will increase as I have a chance to work a little bit more. These preliminary results justify further in vitro experiments. Number of cases might increase in years to come in certain regions of the world, like Canada, if it's transmitted by ticks, because of climate change brings favorable condition for tick population establishment in Canada. So I am really glad that I have been invited here to this conference, and I will bring back the knowledge I gather here with me back to Canada. Before I finish my presentation, I will let you into future studies we have planned. First, we want to use next-generation sequencing to perform microbiome analysis. With this, we'll be able to know all the bacteria that are infecting Morgellons patients that have Lyme versus Lyme-only patient versus healthy controls. I have talked a lot about epigenetics, but we are also interested in looking at the transcriptome, which is the collection of RNA, and it's controlled by epigenetics. RNA is the intermediate between DNA that is imprisoned in the nucleus of the cell and the protein, which are synthesized in the cytoplasm of the cell. Secondly, I will examine the epigenetic status of keratin and collagen using bisulfite sequencing, I have a hard time pronouncing that one, and chromatin immunoprecipitation, or CHIP. And I will explain those two techniques on my next slide. Lastly, I would like to create a reporter gene which could be used to visually detect an upregulation of transcription for keratin and collagen, as would be expected from Morgellon filament formation. And this will be able to see it light up when we infect cells with different combinations of bacterial infections, co-infections. And if we're lucky, we might even be able to produce the fiber or the filament of Morgellons in cell culture. That would be really interesting, but I don't know if that's going to work. As there are lots of types of keratin and collagen, we will have to do mass spec, which is a really complicated technique, not really biological, more like chemicals, so I don't know much about it, but I will get to learn about it. So we will use that to determine which type we should use to make the reporter gene. And this is a diagram just to show you what a reporter gene actually looks like. So you have your gene of interest in red, and then right next to it you put a gene of a protein that you can actually visually see. So this is, most of the time they use GFP, which is a protein that lights up in green, but there's other ones too, like luciferase. And so if my protein of interest in red is being produced, the one right after will also be produced. So I'll be able to see if my gene of interest will be expressed. Bisulfite treatment of DNA is mostly used is the method most commonly used for DNA methylation studies. Methylation groups protect the base cytosine. You can see the M there right over the C. It protects it from being transformed into another base after the bisulfite treatment. So you can epigenetically see what's going on just with a simple PCR. I found that was really cool. So you have to do like PCR with the bisulfite treatment and one without, so you can compare the two. For CHIP, the other method I discussed, DNA is cut into pieces, and then we just pick up the methylated DNA with antibodies. I would like to extend a special thanks to my supervisors, Dr. Lloyd from Mount Allison University and Dr. Robichaud from Université de Moncton, as well as Marianne Middleveen, which is on my thesis committee. I would like to thank them for their guidance, their discussion. I also have to thank the Fraser Morgellons Research Fund for their donation, the Charles E. Holman Foundation for their support, and I would like to thank the study participants and these funding agencies for making the study possible. Lastly, I would like to thank University of Moncton. I have a few pictures over there if you want to see what the campus looks like, and New Brunswick Health Research Foundation that provided me with the funds to come to this wonderful conference, because I would have Skyped if it wouldn't be for them. So I'm glad to be here. I will leave you with a quote by Neil deGrasse Tyson. There is no greater education than one that is self-driven. And that was kind of stepping a little bit on what I wanted to say, but more specifically for Morgellons, I didn't find really anything that suited what I wanted, so I kind of made up my own little quote. What's great about science is that it empowers you to find answers when facing controversies, such as we do in with Morgellons disease. Here are my references. Thank you very much, everyone, for your time, attention.